Today we're going to be talking about Chapter 2, Sections 4 and 5, which is all about chemical reactions and enzymes. Before we get started, check to make sure that you have the corresponding Power Notes sheet. It should look like this, and there are places for you to keep track of all the things that we talk about as we go through this presentation, to include things like drawing the graphs. This will give you great practice as you try to learn this new material. So we need to begin here by talking about what exactly a chemical reaction is, because a chemical reaction does have specific meaning in science, and it's going to be any process that changes or transforms one set of chemicals into another. And what that means is the chemical bonds are changing somehow between what you start out with and what you end up with at the end of the reaction. The parts of the reaction, what you start with, we call those the substrates, or in chemistry class, they would just be called the reactants. As we discuss enzymes, most of the time we're going to call our reactants substrates. That's a very enzyme-specific term. And then what you get out of a reaction at the end, what is produced, is known as the product. So you always can tell reactants versus products if you think about what is reacting or entering the reaction, and then what is produced or what comes out at the end. So in this example, we have a simple combustion reaction of hydrogen gas, which is H2, with oxygen gas, O2. And if you combine these two things together with enough energy, such as in the form of a flame, they actually will react and produce water, H2O. Now we know this is a chemical reaction because we're starting with one set of molecules, H2 and O2, and they are reacting to make two molecules of H2O. That is a different chemical formula, and it's a different chemical compound representing by the changing of the chemical bonds. Now remember, these chemical bonds that we're talking about here are the covalent bonds that are forming between atoms. In the hydrogen, the H2, there is a single covalent bond between those two atoms of hydrogen. And then in the oxygen, there's actually a double covalent bond between those two atoms of oxygen. Whereas in the water, we have a single bond between OH here, another single bond between OH here, and then the same thing in the second water molecule. Those are representing the sharing of electrons, and those are the chemical bonds that we're talking about as having changed. Now, as I mentioned in that reaction, we need the input of energy. So it's important to consider the energy of all of these reactions that we're going to be learning. Activation energy is defined as the amount of energy you have to put into a reaction in order to make it take place. A chemical reaction isn't just going to happen on its own. It needs some input of energy in order to get it started. So you will often see this in the form of a graph like what you have here. You have some starting substrate, you have to input energy into that substrate that makes it unstable, so the bonds start to break and rearrange themselves, and then ultimately form this product here at the end. So this initial hill of energy represents activation energy. You can think of it as this analogy here of frogs jumping over this rock wall to get into this lower pond over here. They have to input energy to clear that wall before they can land in the second pond. So when we are learning about chemical reactions, they're going to fall into two separate categories. We have anabolic reactions, which are the building of large molecules from smaller ones. And we also have catabolic reactions, which are the breaking down of large molecules into the smaller ones. If you've ever heard of anabolic steroids, those are steroids that people take to build muscle. So anabolic is a building process versus catabolic. I tend to think of a cat, an actual fluffy cat with its claws. What do? They tear things up, they shred things. So a catabolic reaction is going to be breaking things down. If we compare what these two would look like on an energy diagram, like what you see here, anabolic reactions are always going to require the large input of energy. right? Things don't just build themselves. You have to put a lot of energy into the starting materials in order to build them into something larger. Versus a catabolic reaction, there's already some energy stored in that large molecule, and you still have to put in some activation energy to make it start to break down, but you actually release a lot more energy than you had to put in. So if we are looking at anabolic and catabolic reactions in terms of their energetics and whether energy is required or absorbed, or if that energy is being released, we have new terms that we would use. And those terms you see on the screen would be endergonic for the anabolic reaction. Ender means into. Endergonic, that second part, gonic, refers to energy. So this would be energy that is being absorbed by those reactants in order to turn into products. And then the term for the catabolic reaction, if we focus on what's happening to the energy, is exergonic. Exer means outer. So this would happen whenever you have energy being released during a chemical reaction. So there's a couple of examples down at the bottom here of what types of reactions these might be in a living thing. We have glucose and fructose. If we are to combine those two together and make sucrose, which is a larger molecule, that would be an anabolic reaction, and it requires the input of energy, so we would also categorize that as endergonic. Whereas oxygen and glucose, if you break down glucose into carbon dioxide and water, that releases energy. This is cellular respiration. This is how we get most of our energy needs on a daily basis. This energy releasing process is important as we are breaking down large molecules. We break down that glucose in order to release its stored energy. So we also need to talk about the rate of chemical reactions, because chemical reactions have to happen quickly enough to be useful in an organism. And some of the things that can affect how quickly a chemical reaction takes place would be if there's any catalysts present. A catalyst is a thing that speeds up a reaction. In biology, enzymes are biological catalysts. We also need to consider, are there things like inhibitors? Just like the name says, if you inhibit something, you're stopping it or preventing it. There are molecules that inhibit chemical reactions. Sometimes that's a good thing. Sometimes you don't want a chemical reaction to take place. Temperature is another factor that can affect the rate of a chemical reaction, as well as pH. And then also the relative concentrations of your substrates, of the catalysts, or the inhibitors, depending on how many of those molecules you have in the solution, that can also impact the rate of your chemical reaction. Now that we know about chemical reactions, let's look at the biological molecules responsible for making them happen, and that would be enzymes. When we are talking about enzymes, it's pretty safe to assume that we're talking about a protein. There are some rare exceptions of enzymes that are called ribozymes, but that's beyond the scope of what we will be learning about in this course. So most of the time an enzyme is going to be a protein like what you see in this picture here. And the picture does a really good job of showing you some of that structure of a protein, how it will coil up. This would be called an alpha helix. Other proteins are folded over into what we know as beta sheets. And that folded structure of the protein is what gives it its particular function. So remember that proteins being made out of amino acids, there's 20 different amino acids. They all can have slightly different chemical properties, and that is what dictates how the protein will fold. So as that string of amino acids that you see right here, as those amino acids start to interact with each other, some of them become attracted to each other. For instance, if they're both polar, or they're attracted to the water that's inside of the cell. And then other amino acids would be nonpolar, and they would be pushed together with the other nonpolar amino acids. So ultimately, you get a folded shape, more like what you see on the right here. And this shape right here is enough to make an enzyme, to make a structure that has a shape that can interact with other molecules. So the shape of the enzyme is really important because that dictates what substrate it can interact with. Remember, the substrate in an enzyme reaction is our reactant, it's what you start with. And the way it reacts with an enzyme is it has to fit into a very specific location, a part of the enzyme that's called the active site. The active site, you can see here in this diagram really well, is a shape that is very specific to one particular substrate, which is shown in a different color so that you can see how it stands out here. And the active site is the place where the substrate will bind. It's where the chemical reaction will be taking place, and that is ultimately what is going to be releasing your product at the end of the reaction. So when we're talking about enzymes, you will hear a lot the term substrate specific. And again, it goes back to that shape, meaning the shape of the enzyme is very specific to a certain type of substrate. Enzymes generally only work on one type of substrate. They do not work on lots of different things, so they can catalyze very specific reactions. Now, the good thing is an enzyme is not used up during a chemical reaction. So even though your body has to make a lot of different kinds of enzymes for all of its chemical reactions, it can reuse them over and over again because they are not destroyed in the process of that reaction. It's really helpful to think about an enzyme and its substrate as a lock and a key. This is a really common analogy that's very helpful in a basic understanding of enzymes. If we think of the lock as being the enzyme, and the keyhole in that lock is like the active site of an enzyme, and then the key would be the substrate or the reactant that is fitting into that keyhole or the active site. Now, this is how scientists understood enzymes to work for a long time. But actually, now that we know more about how enzymes work, we have more sophisticated technology, we know that it's a little bit more complex than that. It's not quite as static as a lock that's very rigid and unable to change shape. So what we're going to talk about next is called the induced fit model. This is really a better way to describe how enzymes work. The lock and key analogy works for a basic understanding, but to be more true to what actually happens, we use the induced fit model. And the main difference here is that in the induced fit model, some enzymes, most or all enzymes, actually have a slight shape change as that substrate is binding. And the enzyme can actually shift position or change shape during the reaction, and that's part of how the enzyme is able to make the reaction take place. But the key for the induced fit model is that at the end of the reaction, that enzyme has returned to its original shape. So if you just look at the starting and the ending point of that enzyme, it doesn't look like there's been any change at all. But during the course of the reaction, there are definitely slight shape changes of the molecule as it's working. Now, before we go through this slide, I do want to encourage you to resist the urge to just write down all of the words that you see on the screen. You probably are looking at this on your Power Notes sheet. This is on the second page, and you see this diagram and you see all of this blank space. Don't worry about writing down all of the words. This is a textbook illustration. It's very wordy. As I go through it, we'll summarize what the most important parts are, and that's what I would recommend writing down on your note sheet. So we're going to start up here at step number 1, and you can probably tell what this is because you see the substrates and you see the arrow joining in to that enzyme there. This is going to be the substrates binding to the enzyme. That's it. That's all you have to write down. You don't have to write down all of what it says up here. Our first step in an enzyme to catalyze a reaction is it has to bind the substrates in the active site. Our next step is that those substrates, they can't just bounce into the active site and then leave. They have to be held there. And that's where some of that biochemistry comes back into play. Hydrogen bonding or ionic bonding can happen between the enzyme and the substrate, which allows the substrate to be held into the correct position while the chemical reaction takes place. Now, in step 3, the entire point of this arrow is to say that the enzyme helps turn our substrate into something new. And this is giving you 4 distinct ways that would happen. I am not going to be asking you on a test what those specific things are. However, whenever we talk about enzymes, I always get questions during class about how an enzyme can actually perform its role. So some of the ways that it can lower that activation energy are shown here. It could act as a template for the substrate orientation, meaning it's holding the substrate in a particular position to make the bond form. It can put stress on the substrate, meaning it could bend it or twist it in a certain way to make a bond more likely to break. It could provide a microenvironment. It could be slightly acidic inside of the active site, which helps a reaction take place. Or it could participate directly in the catalytic reaction, meaning a part of the substrate might actually react with and interact with that enzyme as a part of the chemical reaction. We see this a lot with phosphate groups being added or removed from molecules. All right, step 4 is going to be the conversion of our substrate into product. We can tell that it's now product instead of substrate because the shape and color have changed. Instead of having 2 horizontal pieces, we now have 2 vertical pieces. That represents the bonds breaking here between the top half and the bottom half, and the bonds reforming here in a new pattern. So then we have the products released. When the products change shape and change into a new chemical compound, they are less attracted to that active site and they are able to leave the enzyme. And so finally in step 6, now we have the active site is available for new substrates. So this whole process can start over again. Now let's talk about what happens if enzymes are inhibited. If you inhibit something, you are preventing it from happening. And there are molecules in living cells that inhibit reactions. Sometimes you don't want a reaction to continue indefinitely. Sometimes you want it to have a definite endpoint. So enzyme inhibitors are going to slow down reaction rates. One example of an inhibitor would be cyanide. I'm not going to ask you to know the details of this process, but again, I think it's helpful in understanding if you know a specific biological example. So cyanide is a poison you've probably heard about before. And what it does is it's an inhibitor for an enzyme that's necessary in cellular respiration. So when cyanide, which you can see here is this red dot, when the cyanide interacts with this enzyme, the purple thing is the enzyme, it actually binds to that enzyme and prevents the enzyme from doing its job. If the enzyme can't do its job, your cells can't make ATP, and without ATP, we would die. And so that's why cyanide is a very effective poison, because it's actually an inhibitor of a super important enzyme in the making of ATP. Now, the function of enzymes are going to be to function as biological catalysts. A catalyst, that may be a term you're unfamiliar with, it's simply something that can speed up the rate of a chemical reaction. So that's exactly what enzymes do, and they're biologically produced, so we call them biological catalysts. The way that an enzyme functions, no matter what it's doing, if it's holding the substrate in a position, or if it's putting stress on a substrate, or if it's creating that microenvironment, whatever the action is of the enzyme, the result is always to lower the activation energy. Remember, we talked about activation energy on that energy diagram earlier in this presentation. If you lower the amount of activation energy required for a chemical reaction, you're going to make it easier for that reaction to occur. So let's revisit that energy diagram. And you have this on your note sheet. If you look in your Power Notes, you have this diagram right here. Notice that all of the arrows are blank. I would label every single one of those arrows and just make a note of what we talk about here as we look at this energy diagram. This is probably one of the most important graphs to understand about all of enzymes. There's so much information contained in this one graph, and this information always shows up on the end of course test. So we have some labels here. We see a red line with this really sharp spike in activation energy, a tall amount of energy right there, versus if we think of this one as a shorter hill that has to be overcome. An enzyme would lower activation energy so that we only need this much energy in order to make that reaction happen, as opposed to this much energy that we see with the red line. The reactants start out the same for both the red line and the black line. The products end up the same for both the red line and the black line. The only thing that's different is the height of this activation energy hill right there at the beginning. So again, without an enzyme, it requires more activation energy. With an enzyme, it would require less energy. Don't worry about these abbreviations here. EA just means energy of activation. I will normally just call it activation energy in all of my class materials, but this came from a source that used that particular abbreviation. And delta G is just an abbreviation that talks about energy in a reaction. So this, again, don't worry about that delta G part in that diagram. You should understand, though, that energy, the difference between the amount of energy that was stored in the reactants and the amount of energy now stored in the products, that amount of energy has decreased. So this would represent energy that has been released from a chemical reaction. So this would be an exergonic reaction that resulted in the release of energy. The way that an enzyme works is very much dependent on its shape. And we've talked a little bit about the shape of proteins already, but I do want to mention that it is possible for proteins, specifically enzymes, to have a shape change, and that is called being denatured. Anytime a protein, even a nucleic acid, if it changes its shape, it has undergone the process of denaturation. And if it is denatured, an enzyme is not going to function correctly and probably not going to function at all. So we have what a normal protein should look like on the left. And if it becomes denatured, you can see that it loses all of that specific structure, and now it has a completely different shape, so it's not going to fit with its substrate anymore or be able to catalyze its reaction. Now, there are two major ways to denature a protein that you will normally run across in this class. The first is to expose it to high temperature, not low temperature. You can freeze something and that doesn't denature the protein, but high temperature definitely does. The other way that you can denature a protein is expose it to a pH that's outside of its optimal range. That can either be on the acidic side, which is a low pH, or it could be too basic, which would be a high pH. So you have two pictures here of examples of denatured proteins. There's a lot of protein in egg, and so if you cook an egg, you're exposing it to high temperature, and that denatures the protein. You can tell because it changes texture and it changes taste. The picture on the right is ceviche, which is a raw seafood dish, but it looks like the seafood in this dish has been cooked. It has the same texture of something that has been cooked. But instead of using heat to denature the protein, you are actually using an acid, a combination of lime juice and lemon juice, other citruses that are very acidic. If you soak the fish in an acidic solution, that will denature the protein of the fish, and it will be similar to cooking it. If you look at this graph here, this is a really good way to think about the effect of temperature on a chemical reaction, because you may be thinking that, well, for a chemical reaction in general, if we raise the temperature, it happens faster. And that is true. But remember that we're talking about biological organisms. So there is a point when the temperature gets so high that it causes damage to that biological organism, and that chemical reaction can no longer happen because the enzyme that was allowing it to happen has been denatured and is no longer functional. So here we have a graph which is a really interesting comparison of enzymes that we have in our bodies, and it shows you how different enzymes have different optimal temperatures as well as optimal pH values. So in the top graph, we're looking at temperature. For a human, our human body temperature is around 37 degrees Celsius. So all of the enzymes that function in the human body are going to have an optimum rate somewhere around that 37 degree mark in terms of temperature, because through thousands of years, millions of years of evolution, that is the condition that they have been optimized to work in. Now you can notice this other curve over here has a peak much closer to 80 degrees. That would be way too hot for human enzymes, but heat tolerant bacteria, such as bacteria that would live in hot springs, their enzymes need to function at a high rate of temperature. So that means that as they have evolved enzymes that are able to function at a high temperature. And if we were to put the enzymes from those bacteria into a human, there would be very low reaction rate at all with those enzymes. And then we can see the pH graph. Here is the curve for pepsin. Pepsin is an enzyme that breaks down protein, and it's found in your stomach. So it makes sense that it would have an optimum pH of around 2, which is very acidic. Trypsin, however, is a digestive enzyme that's located in your small intestine, which actually has a slightly basic pH. So you see its optimum pH value is closer to 8. And this is not something that I'm asking you to memorize specific temperatures or enzymes or pH values, but it's a really good example of how different enzymes are very specific to the reaction that they are catalyzing and to the environmental conditions where they're normally found. So the last thing that I want to leave you with here is just an opportunity to do a little self-quiz. This is from your textbook, and go through, look at these questions, find a place somewhere on your note sheet just to jot down your answers from 1 through 5, and we will discuss these when we come into class and discuss enzymes. So hopefully you found this to be helpful, and I will see you in class.